application in osmosis, we're going to see conditions like overhydration or dehydration. And what's going to happen in these circumstances is that water will move in and out of a red blood cell. All right. And when that does that, it can affect the volume of the cell as well as the function of the cell. Okay, so you have a nice little chart here. This would be a good chart to memorize, and then to be able to visualize these. You may want to draw some pictures of red blood cells, and some arrows and water molecules if that's helpful to you. I know a lot of students find drawing very, very useful. All right, what you need to know about this, though, when you start to work on these problems for your exam, let's look at the type of solution. All right, when we talk about the type of solution, we're talking about the type of solution in the environment. All right, the concentration of water in the environment. Then we have the description of what this solution is. Examples, if you look at this part, this is going to be the IV fluids that fit that description of solution. All right, and then this will talk about the effect on the cells. All right, so that would be important to know. So now let's look at how this environment will impact the volume of the cell. All right, so first here we're looking at a normal red blood cell. This is representing an isotonic environment. Okay, these are all little water molecules. There's water molecules on the outside and there's water molecules on the inside. And here's your cell membrane. When those water molecules on the outside are equal with the water molecules on the inside, we say this is isotonic environment. That means that it's iso means same, so it's going to be the same on both sides of that cell membrane. All right, and then in this case, you're going to have water moving back and forth across this membrane, but what's happening is there's no net movement of water. So that means that there's no movement in where it stays in or movement out where it stays out, where it will impact the volume of the cell. It just keeps moving back and forth because that's typical of what happens. All right, and so this is what we want, okay, in most cases. All right, so let's look at B here. What we have here and what we're seeing here is we have low water concentration on the outside of the cell, high water concentration on the inside of the cell. Okay. In other words, we have, if this is low water concentration, that means that it's going to be high solute concentration on the outside and low water concentration on the inside. And so what's going to happen in this case, if this is high solute concentration out here, we call this a hypertonic environment. And in this case, the water is going to want to move from the inside of the cell to the outside environment. And when it does that, it's going to cause this red blood cell to shrink. Okay. And another term for that cell shrinkage is going to be crenation. All right. And then the opposite happens in a hypotonic environment. So remember, this is water concentration, high water concentration on the outside, but high water concentration means low solute concentration on the outside. Low water concentration on the inside means high solute concentration on the inside. Water wants to dilute, so the water is going to move from the outside to the inside of the cell, and it's going to cause this cell to blow up, to swell, and eventually it can burst. And we call that bursting hemolysis, Hemolylsis. All right, lysis means to break open. All right, so this is what we have to consider when we give IV fluids. All right, ideally, in most cases, except for certain situations, we're going to give an isotonic solution for the purpose of preventing these fluid shifts within the red blood cell.